Hello, my name is Juan Matute. I am an neonatologist at MGH, and I want to share with you our work single-cell immunophenotyping of the fetal immune response to maternal SARS-CoV-2 infection. A lot of pregnant women are getting infected with SARS-CoV-2. Fortunately, vertical transmission and neonatal infection is rare. However, a lot of babies are being exposed in utero to the potential effect of COVID-19 infection in mothers, including something called maternal immune activation. The effects of this exposure on the fetal immune system are unknown. In order to address this question, we did a case control study in which we compare the fetal immune system of infants born to mothers infected with the virus in the third trimester and infants born to mothers without infection. None of the infants had vertical transmission. Then we looked in cord blood at multiple genes in multiple cells at single-cell level resolution using single-cell RNA-seq. In this graph, each dot is a cell. We identify several cells through their transcriptome that cluster in non-cell populations in cord blood. We did not find differences in cell composition between cases and controls. However, thousands of genes were differentially expressed in cases and controls as shown in the graph, where the x-axis represents different cell clusters and the y-axis is a measurement of gene expression. Each dot is a gene, and colored dots are those differentially expressed between cases and controls. As an example, we found increased expression of interferon-stimulated genes in cord blood monocytes as shown in the bubble plot on the slide. In the x-axis are different monocyte types, and in the y-axis interferon-stimulated genes. In red are those genes upregulated in cases, and in blue the genes upregulated in controls. There was a variable upregulation of interferon-stimulated genes in cases, which suggests potential exposure to type 1 interferon of cases compared to control before birth. Type 1 interferon is induced by COVID-19 in mothers. This is just one example. We found several transcriptional changes in each leukocyte population we evaluated, suggestive of fetal immune activation secondary to maternal SARS-CoV-2 infection in the absence of vertical transmission. Further studies are required to understand the long-term effects of fetal immune activation on infants born to mothers infected with COVID-19 during pregnancy, even without vertical transmission.